I'm really happy to introduce Hooman. I would certainly wish that he was here in Madison with us. Um, I've known Hooman for, I don't know how long, maybe 20 years.
More.
More than that? Don't say that. Anyway, he got all of his education at UCLA undergrad, and then he worked with Jake Lucis, and he is a very, very broadly based geneticist. He has a very deep understanding of mouse genetics and a very deep understanding of human genetics, and very elegantly uses both to try to understand cardiometabolic diseases. And so he's done quite a lot of work on the genetics of lipid and lipoprotein metabolism. That was really the early work that he did with Jake. And then progressing with the field, I mean, it was really around that time that the field got much more connected with the role of inflammatory pathways in atherosclerosis. And Hooman really got involved with studying lipids that are pro-inflammatory, like eicosanoids. And he was really interested in studying, for example, the 5-lipoxygenase pathway and its role in atherosclerosis. And then subsequent to that, and still to the present, he's been involved in studying gene-diet interactions. So he's very interested in environmental effects, including things like air pollution and their role. And he's also been studying respiratory phenotypes, which I think may have evolved from that. And so he does quite a lot of work on gene by environment interactions in metabolic diseases. This means atherosclerosis, but he also has a really deep interest in fatty liver. Hooman has a really wonderful phenotype, which is pathological optimism.
That's true.
And I like that a lot about him. One time I was with him in a car to some meeting, and he said that he had, in the last several years, submitted, and I can't remember the number, but it was an enormous number of grants, like 20 grants or something like that. And they were all turned down. And he said that with the most cheerful tone. And I thought, wow, that I need that gene. I need to acquire that trait, because I'm devastated if I have one grant turned down. So and he's so resilient. He just says, the next day I just write a new grant. And eventually I know it'll get funded. So he's got that. And, that's a very, very good phenotype to have if you're going to be in science, because a lot of ideas flame out, a lot of great hypotheses. The mice have the last word. They really do. They tell us. And it's heartbreaking sometimes when you get a result and it's not the one you wanted. Okay, so with that, Hooman is going to tell us, what was the title here? Leveraging, his title is Leveraging Large Population-Based Biobanks to Investigate the Genetic Architecture of Coronary Artery Disease. And I'm sure there's even more in there. He'll pack it in, I'm sure. Hooman.
Alan, thank you so much for the kind words. Yeah, I guess I'm an eternal optimist. And it's funny you bring it up, as I'm going to talk to you today. I'm going to sort of show you some of that, I guess, because I know there's a lot of students on, and I just wanted to kind of give them a little bit of like what goes on behind the scenes to getting something published that you just don't necessarily hear in talks. Like people just get up there and say, here's what we did and did it, and here's the paper. But I'm going to sort of show you exactly what you described, some of the trials and tribulations of what we went through to get this story out. That's going to be the basis of my talk. And as you were telling me, I think I need to sort of tighten the net. I think I'm doing way too many things as you described. So anyway, and I also want to thank Aki and Jerry and all the others for inviting me. I really wish I was there. because it's nice to be back in person. I was at a seminar myself last week in person. It was great. So Zoom is convenient, but it just doesn't still, I think, replace the face-to-face interactions, but I'll do my best. I'm not sure. Oh, there we go. Share screen. Okay. So can you guys see my screen?
We see your car.
Yeah, that's also another optimistic thing. It's not my car, but maybe one day we'll see. How about this? Can you see this?
Didn't I tell you about the honorarium for this talk?
No, well, maybe if it's a BMW, great. Okay, can you guys see my screen now?
Yes,
Okay, it's in slideshow mode.
it is.
Great. Okay, so then, do you like to see the mouse cursor, or do you want me to actually go to a laser pointer? Maybe the
mouse is great.
mouse is great. Okay.
Yeah.
All right. Okay. All right. So as Alan mentioned, we kind of use an integrative approach, and we like to obviously do human genetic studies. We like to identify the genetic risk factors, characterize those variants, maybe sort of get a better understanding of the disease, with the ultimate goal of maybe developing novel or therapeutic sort of nutritional strategies, and of course integrating multiple levels of data, either at the transcriptomic, proteomic, metabolomic, even methylomic, and microbiome these days levels, helps us to sort of do this systems genetics kind of studies. And of course, as Alan mentioned, I came out of Jake Luce's lab, which is a mouse genetics lab. So we're comfortable doing mouse genetics, and I kind of view the mouse as another sort of level of data that one can integrate with human. So if you find something in human, you can go to mouse. If you find something in mouse, you can come to human. If you do metabolomics in mouse, you can do metabolomics in human. So it's really nice to, I think, to be able to integrate. And of course, you can do a lot of functional studies in mice that you just can't do in humans. So we kind of like to play around in this sort of a playground. Now, most of our work has been on atherosclerosis, and I'm not sure what the background of most people is. So I just wanted to kind of briefly go over what the pathogenesis of atherosclerosis as we understand it is. And some of this is going to be relevant when I come to the functional work that we've done with this new gene that we found. So atherosclerosis begins with the deposition of cholesterol, which is of course because you have too much of it circulating in the blood, into the subendothelial space. So this is like the space directly beneath the endothelial cell layer of the coronary arteries and other sort of like arteries. And what that does is, because the LDL gets trapped, it gets oxidized, and that causes monocytes to kind of infiltrate in, and they start to engulf this oxidized LDL as a means of sort of like sequestering it and trying to like reverse this, what they believe is like a foreign sort of agent, I guess you could say. And that causes them to turn into foam cells, which become really cholesterol engorged macrophages that grow and grow. This kind of then progresses further, and you eventually get more inflammation, more oxidation. You get sort of like smooth muscle cells proliferating in. The core of this plaque that's developing becomes necrotic. And in some percent, not in all cases, but in some percent of cases, the plaque can rupture because it becomes unstable, either because the necrotic core is too high, or you get sort of like metalloproteinases that might degrade the plaque, et cetera. And once you get a plaque rupture, that's what causes a thrombus, because you get the platelets that aggregate and basically cuts off blood circulation. And so, of course, that's the cause of MI and stroke, and what most cardiologists, I think, would be really concerned with, because that's what causes sort of like a fatal MI or a sudden death, et cetera. You can live with coronary plaque as long as blood is still flowing past the occlusion. But once blood is cut off, that's when you get the problem. So that's just a brief overview of like what the atherosclerotic process is. And of course, from a genetics perspective, and I'm going back to the paper that came out in 2015 from a large GWAS that was done, people have tried to investigate this pathological disease using genetics. Of course, there's also epidemiology, there's functional analysis, but from a genetics point of view, at the time that this paper came out, which was from the Cardiogram Consortium, it involved something like 185,000 cases and controls, and they used like somewhere upwards of like 7 million SNPs or so. But the bottom line was at the end, there were maybe about 100 or so loci at the time that were associated with coronary artery disease. So then when you take those variants plus, what else has been done since then, including exome sequencing and all the epi stuff, et cetera, sort of the picture of the genetic architecture of CAD that one gets is that there's a lot of evidence for common variants predisposing to the development of atherosclerosis and plaque rupture. There's also some evidence, although less so, that rare variants can also predispose. The contribution of common variants, at least at this point, is more than the rare variants, but people still believe that a lot of the missing heritability that common variants can't explain is hidden in the rare variants. And that's why sequencing studies are ongoing. As Alan mentioned, there's also, of course, environmental factors. Diet is probably the major one, along with smoking, but clearly air pollution and other exposures can also predispose. And because of that, you probably have a lot of gene environment interactions that contribute. But of course, these have been very difficult to identify. And a lot of progress in this area really hasn't occurred yet. I, being the optimist that Alan mentioned, as I'm going to mention, with these large biobanks that are available, maybe we can make some inroads in this. But if you take all of these sort of genetic mechanisms or gene environment mechanisms and you ask how do they predispose to atherosclerosis, you see the usual suspects: lipids, blood pressure, and inflammation, thrombosis. But clearly there are, and over half of the loci that have been identified for heart disease, whether rare or common, appear to influence risk through unknown mechanisms. So even though we know something about the biology, as I showed here in this slide over here, there is also a lot that we don't know that has been shown to us by the genetic studies. So if you look at all the loci, most of them do not affect, LDL or, hypertension and things like that. So we really don't know a lot about the pathology, or as much as we should. So we, as the GWASs were coming out over the last 10 years or 15 years or so, we basically had one simple goal. Can we discover new genes and pathways for coronary artery disease and myocardial infarction? It's pretty simple. Now, you can do this either with the outcomes themselves. You can look at it with like the metabolomics level. If there's metabolites that are associated with the disease, can you then identify the genes for the metabolite and by extension find new genes for disease, et cetera? But just to sort of keep it very simple, we thought, well, can we find genes for just heart disease and MI? And, of course, you compete with the big consortiums, or you join the consortiums, but can we find a niche where we can make a contribution? Now, with the consortiums that were available, that was great. And of course, they've published their papers and their summary statistics are available. But then like about 5-6 years ago, and more actually, the data from the UK Biobank resource became publicly available. Now, the UK Biobank, it's a population-based cohort. It's a prospective cohort, meaning that they're accumulating data and following up these individuals. But it's 500,000 people from across the United Kingdom that were recruited between 2006 and 2010, who were between the ages of 40 and 69. So, middle-aged to older adults, 500,000 from across the UK, including Scotland and Wales. The reason why 500,000 people can participate in such a large study is because of nationalized healthcare in the UK. So all of these individuals' healthcare data is in a database, and of course they're willing to participate. So consenting individuals to using their clinical data, plus them providing a blood sample for clinical measurements, biomarker measurements in blood and DNA, allows genetic studies or systems studies to be done on even a larger scale. And of course, you can then combine the results from the UK Biobank with other existing publicly available data to increase sample size even more. So this is just a brief overview of the things that they have done in the UK Biobank. They have obviously a lot of the socio-demographic lifestyle factors that they get at an interview. They've done imaging such as MRI on the brain and on like the thoracic imaging and abdominal imaging. It's not in all 500,000 people, but it's in like about 40,000 individuals. You have death records, electronic health records. They have, like I mentioned, genomics. It was GWAS based. Then they've done exome sequencing on all the individuals. And I believe now they're halfway through or in the midst of whole genome sequencing 500,000 people. And all of this data is publicly available. You can apply and get this data if you want. They also have a bunch of biomarkers like cholesterol measures, et cetera, and recently released metabolomics data. So it's really a treasure trove resource that one can use to investigate not just cardiometabolic, but any disease outcome for which they have data. So at the time that the UK Biobank was coming online and one can get access to it, as well as the cardiogram big meta-analysis that I mentioned to you, we thought, well, is there a way that we can sort of like combine the data together and basically essentially increase the sample size and find new genes for heart disease? So this is what I call the search for new CAD genes 1.0. So this was in mid 2017, just to give you a sort of an idea of when we started this. This is about going back 5 years. So we submitted an application to the UK Biobank in the summer of that year, 2017, and we were waiting to get approved. And then in the fall of 2017, Ben Neal, who's, I believe he's at the Broad now, published a blog really where they had already GWAS'd 2,000 phenotypes in the UK Biobank and posted it on their website. So essentially what we wanted to do, this group had done. So we downloaded that data and we said, well, instead of us analyzing it while waiting for the UK Biobank, why don't we take that data and just basically meta-analyze it with cardiogram and find new genes that way, kind of like a shortcut. So as we were going through this, we found about a dozen loci shown here. This is the results of our meta-analysis that we did with the Ben Neal UK Biobank results and the results from the Cardiogram Consortium, the publicly available summary statistics. We put it all together for about 500,000 people. And what you see here in the red is about a dozen loci that were identified for heart disease that were not previously known. So we thought that, okay, great. We found like, a dozen or so novel loci. We're kind of like, excited by it. We were trying to get the data organized. This is again, November, December of 2017. And then all of a sudden, in my PubMed search, I see this paper from Pim Vanderhorst, who's also in the UK, where they essentially did exactly what we had were doing at the time. So they had taken the UK Biobank data and meta-analyzed it with cardiogram and found 64 novel loci. And so basically we thought, well, we just got scooped. So what are we going to do? So then we thought, well, there was some evidence in the literature going back 10 years or so that it's not just genes for heart disease, but there could be genes that are specific to the rupture of a plaque. So basically genes that are predisposing to MI, but not necessarily heart disease. And this was a Lancet paper that Murdoch Riley published in 2011, showing that the ABO blood group system on chromosome 9 was specifically associated with heart attack, but not heart disease itself. So because of the fact that, this paper had come out looking for CAD genes and this sort of like data in the literature, we thought, well, why don't we sort of like change our approach? And now maybe there's other genes that specifically predispose to plaque rupture. We know genes that contribute to plaque development, which then go on to affect MI. But are there genes that specifically predispose to plaque rupture once you develop a plaque for whatever other genetic reasons? So it was looking for the progression from the presence of plaque to the rupture of a plaque that we wanted to sort of maybe make a contribution. And again, ABO, if you want to call it, is the positive control locus for this sort of like mechanism, genetic mechanism. So we basically reanalyzed the data that we had from the Ben Neal group plus the cardiogram, because the cardiogram people had also posted their summary statistics for heart attack separate from heart disease. And when we did that, we still had like about half a dozen or so novel loci that were associated with heart attack, and they were not associated with CAD that we knew. So And this is again for the students. So we thought, okay, well, it's not as exciting as it was before, but at least we have still something new that maybe we can sort of contribute to the literature. So we drafted the paper and we sent it off to circulation, and within a month it came back as rejected. This is now October 2018. So this is over a year after we started. So we thought, okay, well, that's fine. You know, some of the comments might have been legitimate. Of course, you sort of disagree with some of the other comments, but we thought, well, they probably just didn't think that it was novel enough given the circulation research paper that was published like less than a year before this. So then what we did was we took into account some of the comments from the reviewers, revised the manuscript in early 2019 and submitted it to Nature Communications. Then we waited and we waited and we waited. We waited 2 months and it came back rejected. So I thought, okay, well, so be it. So Alan, you know, you said that like, you just get rejected and you move on. I mean, the people in the lab were a little bummed. I'm like, you know what? It's okay. You just got to move on. What are we going to do? We're going to give up and cry? We just got to keep going. So what happened in the end, actually, it turned out much better. So what happened is during this whole time that we're analyzing the Ben Neal data, and then, people were criticizing it as saying, well, it's not the right way to analyze UK Biobank data. Our application for the UK Biobank had been approved and we had downloaded all the data ourselves. So I just told people in the lab, I'm like, you know what, forget the Ben Neal data, go back and let's just analyze the UK Biobank data ourselves where we have control of it. And then we can meta-analyze it with cardiogram the way we want to, not the way that it was done by another group. So the shortcut really didn't work. But at least it gave us a sort of like practice in how to do these sort of large scale meta-analysis. So with that, we had search for new MI genes 3.0. So the story is kind of evolving. So then we said, okay, now we're going to carry out our own studies with the UK Biobank. We added some additional cohorts in our study and functional data through new collaborations. So really the study grew quite a bit. And of course, that is manifested in the author list. If you look at the original author list from the first submission to what was eventually published, I think it doubled in the number of people that we included. But that's team science these days when you want to do these large scale human studies. And what we said was we're going to not really look at CAD genes, but we really want to focus on genes for MI. What are the genetic risk factors specifically for MI? And are there any additional ones besides ABO? So after 3 years of work, we finally submitted a new version of the paper to the European Heart Journal in May of 2020. And, said, well, maybe third time's the charm, who knows? But we had our fingers crossed. And as you probably know, it was successful. So now I'm going to tell you of well, why did it become successful? So here's just the big picture of what we did. So as I mentioned, we had data in the UK Biobank ourselves. So we had 17,000 cases with heart attack, 450,000 controls, and about 11 million SNPs. We did a GWAS analysis for MI by itself and found some loci. And the Cardiogram Consortium had posted their GWAS summary statistics for heart disease versus controls as well. So we basically took our analysis and the publicly available summary statistics and did a meta-analysis. And this basically brought us to about 600 and I would say 30 or so thousand cases and controls with 8 million SNPs. In the end, as I'll show you in a second, we found 8 novel loci for heart attack that had not been observed at all for CAD in the past. And then we did some follow-up studies and some replication and functional to eventually land on this gene that we think is an MI-specific genetic risk factor, SLC44A3. So that's just the big picture.
So Hooman,
yeah.
Can I ask you a quick question?
Sure.
So the new genes in the figure you had a little while ago,
yeah,
all had lower lodge scores than the old genes. And so does that mean that this all happened because you were looking at a much bigger sample size, and so you were looking in the weeds, basically?
Exactly. Yeah, Alan. So some of those were probably suggestively associated, and then as you increase the sample size, they get above this magical threshold of 10 to the -8 that everybody sort of expects to see. Now, I know that that's just 5 times 10 to the -8 is basically like a Bonferroni threshold for significance. And there's a lot of stuff in the weeds that's probably truly associated, but people just expect it to be less than 10 to the minus 8 before they believe it or not. So does that answer your question?
Yes, it does.
Yeah. Okay. So that was the big picture, this sort of like flow chart that I showed you. And so let me just show you now the data that we had in here. So as you see, this is the Manhattan plot for the meta-analysis. And what you're seeing with like the orange are those 8 loci that we had found. Again, Alan, this is to your point that some of these are barely like above the threshold, which is the red horizontal line. The blue horizontal line is the suggestive threshold. And you'll see quite a few loci that are in that suggestive range. And who knows, if you add like, you know, more cases and controls, those might actually become significant as well. So we could be missing quite a bit. And now the question was, can we follow up on these 8 loci a little bit and truly demonstrate that maybe they are specific for MI and not maybe for CAD? So one of the first things we did is we took each of the lead SNPs, and those are shown here. These are the loci, and then these are just the nearest genes to the lead SNPs. And we compared the association odds ratios and the association p-values for heart attack compared to coronary artery disease. Remember, this MI is what we actually GWAS. And if you look at all the p-values, they are genome-wide significant because they are all less than 5 times 10 to the minus 8. When you compare the associations with CAD to that with MI, you'll see that the association odds ratios and the p-values are attenuated compared to those for MI, with the exception of perhaps the first locus up here, this AHDC1, which has a 5 times 10 to the minus 8 p-value, as well as this one down here, which is the FHL5, which also has a 1 times 10 to the minus 8 p-value. But the rest would all be not considered significantly associated. So this was the first clue as to maybe these loci are more specific for MI. Of course, this CAD result was based on the meta-analysis. And since we had primary level data in the UK Biobank alone, we thought a better way to compare this sort of association signals was to do it in the UK Biobank itself. Another advantage of that was that we could take individuals with heart disease and then ask just among the individuals with heart disease, what is the association of these loci with MI? Because these comparisons are MI versus control, CAD versus control. But now if you take all the people with CAD and separate them into those with and without MI, you'll get a better evaluation of whether they're associated with plaque rupture or not. So that's the data that's shown here. If you take individuals in just the UK Biobank alone, and you ask people with MI versus controls who don't have MI or CAD or anything, what is the association signals at these 8? You'll see that they're pretty, you know, they're significant. Some of them are more significant than others, but these are the association signals that contributed to the overall meta-analysis. When you take the people who only had heart disease and no history of MI at all, and you ask, are these loci associated with just the presence of plaque itself, you'll see that none of them are associated, with the exception of, again, maybe that first one, AHDC1, which, as I showed you on the previous slide, is the one that was genome-wide significant for heart disease as well in the big meta-analysis. Then the next comparison we did was we said, okay, you take the 16,000 people who only have heart disease and you compare them to the 17,000 people who have heart disease plus an MI, and what is the association of these loci? And you'll see that amongst all individuals who have heart disease, with the exception of the first one and maybe this third one right here, all the other loci do show an association with MI in the presence of heart disease. So this kind of suggests, and if I'm going to go back to this right here, that there are risk factors that are predisposing to plaque rupture in the presence of heart disease. That was like the first sort of like clue. And now of the loci that we had, you could say that maybe 1, 2, 3, 4, 5, 6 of them kind of gave us this pattern. And by the way, we also had, I was actually, let's see, 1, 2, 3, 4, 5 of them. And down here at the bottom, we used ABO as that positive control locus, because if you look at the ABO locus, it is associated with MI, it is not associated with heart disease, and it is associated with MI in the presence of heart disease. If this is the positive control locus, we would expect the other loci to mimic this association pattern. And for the most part, those 5 other loci do, although the associations are not particularly strong. So the next question was, well, can we replicate these associations in additional cohorts? So we went and we collaborated with quite a few people who had angiography-based cohorts. These are individuals who have been recruited into studies based on angiography. So for whatever reason, mostly because of CAD, they were referred to angiography, they had angiography done. And so you definitely know if they have heart disease or not. And then amongst those individuals, you can then stratify them into the presence of heart disease with or without a heart attack. So it's essentially the same comparison that we did down here, but in one set of angiography-based cohorts, that's about 13,000 or so, maybe 14,000 individuals, followed by an additional set of 16 separate cohorts, independent, with about another 13,000. And when you look at those 6 loci that we looked at, that we evaluated in the previous slide, only one locus, this locus on chromosome 1, shows a robust association with MI in the presence of heart disease, both in the first set of angiography-based cohorts and the second. And of course, when you meta-analyze these together, you get a nice p-value of 5 times 6 to the 10 to the minus 8. So that was like exciting, because we thought, well, maybe this SLC locus could be really a truly MI-specific risk factor. And as we were going along and we were in the midst of revising the paper, et cetera, this paper came out in Nature Genetics, which is another biobank. This is now Biobank Japan, similar to the UK Biobank, but not as large. And they had done a big genetic analysis for heart disease in the Japanese, and of course, meta-analyzed it with the UK Biobank and cardiogram. And essentially what they did is they found 8 specific loci for the Japanese. Again, this is for heart disease, not MI. So they didn't really scoop us here, because they were looking at heart disease itself, not specifically MI. But in the end, they expanded beyond that circulation research paper that I showed you that scooped us by adding 43 new loci for a total of 204. So now, as of 2020, there are 200 loci for heart disease itself. But luckily, the SLC locus didn't come up on their radar, because they weren't looking for MI specifically. So we reached out to them and we said, we have this gene, we think it's associated, or we have some loci that we think are specific for MI. Can you analyze your data the way that we have done it and provide us the results for our candidate loci? And they were very willing to help. I will tell you that, in the cardiovascular field, for the most part, people are very collaborative, and it's been very nice. People are, they're very willing to help out. And so it makes it a lot more fun. And so here are the same analyses that I showed you on the previous slides for MI, CAD only, and then MI in the presence of CAD, using data from Biobank Japan. And these are the sample sizes up here. So again, you see, if we look at these loci that we identified, if you look at ABO as the positive control, a very nice association with MI versus controls, no association with just heart disease by itself, and then a nice association with MI in the presence of heart disease. So again, this pattern is exactly what Murdoch-Riley reported in 2011, what we observe in the UK Biobank, and what the Biobank Japan cohort also shows. And of the other loci that we identified, again, the only one that shows the same pattern as ABO is this locus on chromosome 1, where the nearest gene is SLC44A3. A p-value of 10 to the minus 5 for MI versus control, no association with CAD only. And then when you do MI in the presence of CAD, you get a nice association there as well. So we, based on just statistics and genetic analysis in human cohorts, we believe that, this SLC locus represents a novel and MI-specific risk factor. Okay, so what does the locus look like? So here is the lead SNP, RS127-43267. It is actually physically present in this non-coding RNA, which actually turns out to be an antisense to SLC44A3, which is right here. So the antisense is transcribed from right to left on the bottom strand, and an SLC is transcribed on the top strand from left to right. And the SNP is, I guess you could say, is somewhere in the promoter region of SLC. and possibly in the regulatory, or at least the intronic region of this antisense. So, you know, if you just go by nearest gene, that doesn't mean that that's the causal gene. And in looking at the genes that are in this locus, what you'll see down here is factor 3. So factor 3 is a coagulation factor. It's involved in clotting. And because MI and stroke is really a thrombotic phenotype, one would think that, oh, you know what? Factor 3 is actually the biologically relevant gene, not SLC44A3, which at the time that we sort of landed on this locus, there were only like, I think, less than 6 papers on SLC44A3 in the literature. So really nothing was known about SLC44A3, but factor 3 was a great candidate. So as I mentioned, it's the gene that encodes tissue factor. When tissue factor, you know, when you get like a sort of a tear inside the artery or whatever, and tissue factor is exposed, that what starts sort of like the coagulation cascade. So it's crucial in clotting and sort of like stopping bleeding. And what was interesting is that several years prior, there was a GWAS that was done for D-dimer levels, which is a breakdown product of fibrinogen and a commonly used biomarker for coagulation. And there was a SNP that was upstream of factor 3 that was associated with D-dimer levels. So we thought, well, if there's a SNP, if factor 3 SNPs are associated with D-dimer and our MI SNP is affecting MI because of factor 3, then our SNP should also be associated with D-dimer. So we contacted the people who had done the D-dimer GWAS, but our SNP, RS127-43267, was not associated at all with D-dimer, and the D-dimer SNP that was found in that GWAS was not associated with MI. So based on that alone, we kind of thought maybe factor 3 is not the gene that's underlying this association with MI. So then we turned our attention to SLC44A3 and some of the other genes, but I'll tell you more in a second why we focused on 44A3. Really, nothing is known about it except it had been postulated to be a choline-like transporter. And that was simply based on the fact that there is a family of these solute carriers, SLC44A1, 2, 3, 4, and 5. And there was some functional data showing that SLC44A1 was a choline transporter, and that's why they attributed the same function to 44A3, but it hadn't really been shown, and it still hasn't been shown. But the other reason we kind of like turned our attention to 44A3 was that 44A2, which as I mentioned is just another member of this family of transporters, had been linked to venous thromboembolism, which is basically a clot that's formed in the veins instead of the arteries. And so this is like, you know, DVT when they tell you on the plane to get up and walk around, or you wear those compression socks, it's to basically prevent venous thromboembolism. So because 44A2 had been associated with the venous thromboembolism, we thought, well, maybe 44A3 could be associated with an arterial thrombosis, namely MI. That was sort of like the rationale. So what do we know about 44A3? So this is using human gene expression data from GTEx, where they've done basically RNA-seq on a variety of tissues. And if you look at the tissues in which SLC44A3 is expressed, the highest one is salivary gland and colon and kidney. When you go down, you know, really like artery, coronary artery, this is aorta, and where's the coronary? I forgot where it is. Even coronary artery even shown here? Maybe I don't have a coronary.
It's the light pink. It's the light pink.
The light pink. Light pink.
Just go right.
This one?
Yeah,
thank you. Thank you. So it's really in the middle of the pack in the sense of like, you know, how highly it's expressed. That's not a reason to exclude the gene, but it kind of like, you know, gives us an idea of if we think that we have an MI specific genetic risk factor, you would like the gene to be expressed at reasonable levels in the tissue for which you think is the underlying pathological thing, which is the artery wall. But nonetheless, you know, based on GTEx data, we do see some expression. Then we went and we looked at a different cohort called StarNet. Now, StarNet is a cohort of individuals who have undergone bypass surgery, and they've collected a variety of tissues from those individuals while they're undergoing bypass. Namely, they get visceral adipose, a piece of the aorta, because, you know, they kind of trim the thing for the grafting. They get a piece of liver, subcutaneous adipose, mammary artery, because that's what's used often for the grafting, plus a piece of skeletal muscle and blood. And this study was not done in the United States. I don't know if you could ever get approval to take out these kinds of tissues for somebody who's having open heart surgery. So it was done in Europe. I believe it was in Estonia, where I guess they consent very easily. But if you look at the tissue expression pattern of SLC in these tissues, you kind of see it, not super high levels, but, in tissues that could be relevant, namely aorta, possibly liver and adipose. So we had that. Then we looked to see whether the lead SNP yielded an EQTL in these vascular tissues. In StarNet, you see kind of a modest EQTL in aorta and normal mammary artery. It's the C allele, the major allele, is the one that increases risk of MI. So the fact that the C allele is associated with higher expression levels of SLC suggests that there's a positive correlation between SLC and risk of MI, namely more SLC, more MI. If you look at the EQTLs in the aorta and coronary artery from GTEx, you see a similar pattern where individuals who carry one or two C alleles have higher expression than people who have the T allele, again suggesting that more SLC might be more causally associated with MI. And then we did some additional, and this is where collaborations with other groups really help. There was a group, Clint Miller, who's at the University of Virginia, had RNA-seq data in ischemic coronary arteries and normal coronary arteries. And when we look at the expression of SLC, it's elevated in ischemic coronary arteries compared to normal. That also is consistent with maybe SLC increasing risk of MI because it's differentially expressed in arteries that maybe don't have enough blood flow. Then we looked at the expression levels of SLC in endothelial cells after being treated with a pro-inflammatory cytokine IL-1 beta, because if you have this increased expression in the arteries, you'd like to know which cell type is it possibly working through. And the data shown here in panel E suggests that, with the upregulation of SLC in response to a pro-inflammatory cytokine in endothelial cells, maybe endothelial cells are one cell type that could be underlying this MI risk. And then the last panel over here shows an in vitro assay with smooth muscle cells in terms of their migration rate versus expression levels of SLC. And you see a modest sort of inverse relationship between SLC expression and in vitro functional evaluation of smooth muscle cells. All of this really doesn't sort of like say, oh, aha, it is for sure SLC, but it suggests that maybe SLC is doing something in the vascular wall, and that could be predisposing to MI. That's sort of like the working hypothesis that we have. Okay, so now the question was, do we have any clues as to what SLC44A3 could be doing functionally, right? As I mentioned, there wasn't a whole lot known about it. So this goes back to what do we know about 44A2, which back in 2015 was identified as being one of the most strongly associated loci for venous thromboembolism. And because of this GWAS that had been done way back then, two groups had characterized an SLC44A2 knockout to determine whether that could be the basis for its association with VTE in humans. This group here, it was a European group, I believe, showed that mice that don't have 44A2 had increased bleeding time and decreased thrombosis, suggesting that 44A2 in humans actually is positively associated with venous thrombosis, meaning more 44A2 activity or function increases risk of thrombosis because the 44A2 knockout had decreased thrombosis. But they didn't really show a mechanism. And then this paper came out from Charlie Lowenstein's group. This is from, is this 2020? I think it's 2020. I don't have the date here. I believe this was 2020, but they also characterized the 44A2 knockout, but they took it a step further and actually figured out the mechanism for it. And it's related to choline. So the first thing they showed is that the 44A2 knockout has increased bleeding time, therefore decreased risk of thromboembolism. They also demonstrated that the increased bleeding time can be recapitulated through bone marrow transfer, so that it's not because of, like, let's say the vascular wall itself, but it could be something that's mediated through bone marrow derived cells. And of course, they focused on platelets, as I'm going to show you in a second. And then they also showed that reactive oxygen species in response to thrombin incubation in platelets is reduced in the knockout compared to the wild type. So they think that it's affecting platelet function. And one of the reasons, and they to sort of investigate that mechanism a little bit more, they did metabolomics on mitochondria from platelets, and they showed that choline and some choline-derived metabolites, such as dimethylglycine and betaine, are decreased in the mitochondria of the SLC44A2 knockout. That would be consistent with the 44A2 serving as a choline transporter into mitochondria, because 44A2 is a mitochondrial localized carrier. It's actually on the mitochondrial membrane. Then they went and showed by a seahorse that 44A2 knockout mice have decreased mitochondrial function in platelets. So you can see here that in the dark black, they have decreased oxygen consumption. And when you give them choline, even though the wild type mice, it increases their oxygen consumption, the same does not happen in the knockouts. Again, suggesting that it's choline-derived ATP production that is promoting thrombosis. At least that's the goal. That's the idea that they suggested. So here's the model that they went with. SLC44A2 transports choline into the mitochondria. That promotes ATP production, which then leads to platelet activation and thrombosis. And when you don't have 44A2, this does not occur as much, and therefore you decrease thrombosis. So based on this data, we asked ourselves, yeah.
How does choline cause ATP production?
Well, Alan, that's a good question. I should know that answer. I believe it's oxidized to produce it, but I have not actually, believe it or not, I haven't looked at the biochemistry of that, which I'm embarrassed to say.
I don't, this is new to me. And then the other question I have is, do vascular cells make acetylcholine?
Just another really good question. I don't know about that either. Yeah. That's, yeah.
Because doesn't acetylcholine have vascular properties?
In terms of like vasodilation, you mean, or something?
Yeah, yeah.
Does it?
Yeah, I think so.
Huh. Well, yeah, I mean, I don't know. So we can look at, what is the enzyme that makes acetylcholine?
It's an acetyltransferase. I don't remember the name of it, but that was my next question, was in the metabolomic analysis, did you see, was acetylcholine one of the metabolites?
Yeah, this is the Lowenstein data. So they didn't, I don't know if they posted their metabolomics data, but one can go and look. I mean, but again, Alan, this is in platelets. They did it specifically in platelets. So the metabolomics was all done in platelets.
Okay. But
so
you're more interested in, I thought that your interest was more in smooth muscle cells.
Right. But I'm showing you the data that they had with SLC44A2 and platelets, because now we're wondering if 44A3 is doing the same thing as 44A2, except in the vascular wall, right? Can we use the data that they demonstrated for platelet activity in choline transport and mitochondrial function, and then say, okay, 44A3 might be functioning in the same way, except in a different tissue that's more relevant for MI? That's kind of where we're heading. And I'm going to now show you some unpublished data that maybe suggests that could be the case. So okay. Yeah, but that was the rationale for showing you all this data that was published by Lowenstein. So one of the first things we did is we went and said, well, maybe 44A3 is doing the same thing in platelets, and why are we focusing on smooth muscle cells? So we went and looked at data from an RNA-seq paper that had looked at the transcriptomes of platelets in both mouse and human. And when you look at the 5 SLC transporters that are part of this family, in mouse or human, what you see is that 44A2 is robustly expressed in platelets, as Lowenstein demonstrated as well, right? But there are no counts for 44A3 in platelets. So it was nice because at least we said, okay, we're going to rule out platelets as the cell type that 44A3 is predisposing to MI. Okay, it can't be platelets. So could it be really vascular wall? So the next thing we did is we said, okay, where is this lead SNP that we found for MI located? And we used single cell ATAC-seq data that Clint had generated in coronary arteries. And when you look at the location of the lead SNP, you'll see that it's smack dab in the middle of a peak that you see in coronary arteries, smooth muscle cells, fibroblasts from the coronary artery parasites, but really not in macrophages or endothelial cells. And this region is shown to have a long-range interaction with the SLC44A3 promoter, but no other gene in this region, including factor 3, which is way over here on the left. So the lead SNP appears to be potentially regulatory in a sort of region that is interacting with the SLC44A3 promoter in vascular walls, with the largest peak being in smooth muscle cells. So Alan, this gets to me where I like, you know, we're trying to, we think it's actually smooth muscle cells. This is the first piece of data. Okay, then we used single cell RNA-seq data generated from human atherosclerotic plaques. And when you look at SLC expression at the single cell level in atherosclerotic plaques, you'll see that it's really expressed not in all smooth muscle cells, but in a fraction of smooth muscle cells and the derived fibroblasts, et cetera, in the plaque, but not so much in endothelial cells and certainly not in the inflammatory cells. So if we had to pick a cell type that we think SLC44A3 might be functioning in with respect to MI, then we would maybe prioritize smooth muscle cells as one. And this is the reason why I showed you that sort of like pathogenesis slide initially, because smooth muscle cells are clearly important in the sort of the advanced plaque and its destabilization. So maybe it's smooth muscle cells. So then another thing we did is we contacted Jake at UCLA, and I knew that Jake had gene expression data in about 100 inbred strains of mice, the hybrid mouse diversity panel. And we asked a simple question: in 3 relevant heart disease or MI tissues, namely liver, adipose, and aorta, if you take the genes that are most correlated with SLC44A3, so we basically took SLC44A3 expression in liver and correlated it with all the other genes in liver and just took the top 1,000. If you take those top 1,000 most highly correlated genes and you do an unbiased sort of pathway enrichment with ingenuity, what are the pathways that come up? In liver, the top pathway is protein ubiquitination with a p-value of about 10 to the minus 7, and then like, you know, sort of less significance with these other pathways. If you do the same thing in adipose tissue, the top pathways that pop up are mitochondrial dysfunction, glucuronate degradation, and oxidative phosphorylation, although the enrichment p-values are not that great. If you do the same thing in aorta, the top 2 pathways that come up with pretty significant p-values are oxidative phosphorylation and mitochondrial dysfunction. So if you put this together with what Lowenstein showed in platelets with the SLC44A2 knockouts, and what we think our gene or our lead SNP is doing with respect to smooth muscle cells and SLC44A3, the question becomes, could 44A3 be affecting mitochondrial function, but it's just doing it in the aorta, in the vessel wall, and possibly through smooth muscle cells? That's now sort of like the hypothesis that we're moving forward with. And the other thing that Jake had was he had mitochondrial DNA content in liver and adipose. So we simply asked, if you correlate SLC44A3 expression with mitochondrial DNA content in those 2 tissues, what do you see? And so in liver, there's absolutely no correlation between mitochondrial DNA content and 44A3. If you look in adipose, you do see a nice positive correlation. R is 0.55. This is, again, about 100 inbred strains, p-value 10 to the minus 11. And this is consistent with at least mitochondrial dysfunction and oxidative phosphorylation being some of the top most enriched pathways. Now, of course, the big question was, what do you see in the aorta, right? That would have been like the greatest thing. But Jake, unfortunately, they never looked at mitochondrial DNA content in the aorta because they had no reason to. And so this is one thing that we're trying to now address. So how are we addressing this? Well, there is an SLC44A3 knockout that was generated by the International Knockout Mouse Consortium. And it was generated in Germany. So after many months and a giant import payment, it was like $7,000 to import these mice from Germany, just because of you know, their international, you know, et cetera. We got our hands on the knockout, And some people had said because of the way that they generate knockouts, and this is their sort of like diagram of what they used and their vector, et cetera. In the end, after injecting Cree, they said that, you know, it would remove exon 2. So when we got the mouse, we thought, well, we want to validate the fact that this is really a knockout. Some people have told us that, oh yeah, if you get like knockouts from the International Knockout Consortium, you should really characterize the mouse before, like, you know, sort of doing studies with it. Sometimes they're not knockouts. So we basically took the DNA and we amplified the various beginning exons, and we actually got a signal for exon 2 and exon 3, but it turned out that it was exon 4 that was deleted in this knockout, not exon 2. So the next question was, well, does that actually generate a knockout? So we sequenced the cDNA from the wild type allele and from the knockout allele. And it turns out that, you know, when you do this sort of with the Cree recombination, et cetera, you do get an EN2 sequence in place of exon 4, and that leads to a premature stop codon. So we're pretty sure that it's a knockout. And this was further validated by doing RNA-seq in aorta on the wild types, the knockouts, I'm sorry, the heterozygous knockouts and the wild types. So you basically get no SLC expression in the aorta in the knockout. You get some expression in the wild type, and the heterozygotes are in the middle. So we think it's a knockout. Unfortunately, there is no good antibody for this 44A3. We tried several like that were supposedly commercially available and work, but none of them work. So we're stuck kind of assuming that it's a knockout based on expression levels. So then we thought, okay, one of the first things that we can do, because this is such an important question for us, is there a difference or is there a relationship between mitochondrial DNA content and SLC in the aorta? We basically went and characterized the SLC knockout for mitochondrial DNA content in a variety of tissues. So in liver, adipose, and colon, there is no difference in mitochondrial DNA content, but in the aorta, the knockouts have reduced mitochondrial DNA content. That's consistent with the correlation that we saw in adipose and everything that we would predict up until this point. This was done using a mitochondrial DNA content we developed at USC in our lab. So, you know, because it was our first foray into this, you know what I did is I personally took our mice, put them in the car, and I drove to UCLA. And because Jake's technician also does mitochondrial DNA content, I asked Sachan, can we take out aortas in your lab and you do the mitochondrial DNA content using your primers and, you know, all that stuff, your qPCR assay, just to sort of independently validate this. And when we did that, we saw decreased mitochondrial DNA content using completely different primers and in different assays. So we're kind of happy about this, and we think we might be on the right track. And so this is the, you know, the data that we have until this point. Now, of course, it's nice, you know, we think it's SLC44A3, maybe it's related to mitochondria, but there's a whole host of questions that come up. Number one, if you believe the mitochondrial DNA content difference, is it cell type specific? Meaning we've done whole aorta. Do you see decreased mitochondrial content in the smooth muscle cells, in the endothelial cells, in other cell types, or is it just like in one cell type? More importantly, if there is decreased DNA mitochondrial content or DNA content, does that lead to altered mitochondrial function? And if so, is that cell type specific? So do we need to do a seahorse assay in smooth muscle cells, in endothelial cells, or other cell types to figure out what's going on there? And more broadly, if we have, like, let's say the knockout, what other pathways could possibly be involved? And does SLC44A3 regulate other pathways that could also contribute to plaque rupture? At the sort of like global, sort of like physiological recapitulate the human phenotype, does SLC44A3 deficiency affect plaque rupture in mice? Can we model an MI in mouse and demonstrate that 44A3 knockouts have decreased MI, right? Or decreased plaque rupture? And then again, at the broader picture, can we sort of increase our sample size in humans? And again, this is the sort of ping-ponging back and forth that we like to do. Can we find even more genes that are specifically associated with MI in the presence of heart disease? So just in the last few slides, I'm going to tell you about how we're approaching this. There are multiple mice in general, no matter how high you take their cholesterol or how much atherosclerosis they get, they really don't exhibit plaque rupture. That's why it's not a good model for MI. But through various surgical models, by sort of like creating like ligations of certain arteries and stuff, people have suggested that you can actually get ruptured plaques. And this is a paper that came out by Karl-Heinz Peter, he's in Australia, where they do a tandem stenosis model of the carotid artery. And this, I don't know the exact sort of like, physics of the fluid dynamics, et cetera, but by having two tandem stenoses in the very proximal segment right here, before the first stenosis, what you can get is not only do you get plaque buildup, but you actually get evidence of plaque rupture. And that's evidence of like with these thrombi that are here, and you get some blood cells, et cetera. So Karl-Heinz suggests that this surgical model would sort of recapitulate a plaque rupture in humans. That's his idea. And so we've talked to him, and one of the things that we'd like to do is basically do this exact surgical model with our SLC44A3 knockout mice, and predicting that, yes, the knockout would actually have decreased evidence of plaque rupture. So that's how we would like to recapitulate the human phenotype in the mouse. And then the other question about, well, can we find even more genes for heart disease, for plaque rupture or for MI? We thought, well, why don't we actually go and do a GWAS analysis only with people with heart disease, and then stratify them into those with and without MI. It's the same analysis that I showed you specifically for 44A3, but now can we do it on a sort of like a GWAS level? And with all the different cohorts that we've tried to sort of put together, in the end, we can get about 80,000 people with heart disease and MI, and about 86,000 people with only heart disease. And in a subset, we've already done an analysis, and here's the GWAS results for looking for MI only among CAD cases. This is not a GWAS analysis that has been done in humans yet. And when you do that, what you see is, here's ABO on chromosome 9. So we have one positive control. SLC pops up as being significantly associated. You have these other loci, and including this really, really significant one on chromosome 12. So it turns out that this locus, because this analysis includes the Japanese, this locus is because of a SNP that is only present in the Japanese and had been seen for MI in prior studies. So this is really a Japanese sort of specific signal that we're seeing again. This SNP is not present in any Caucasian data sets. And if I go back to the cohorts, really the only, this sort of interim analysis that we've done includes UK Biobank Japan, and some of the angiography-based cohorts that are of European ancestry. So this is really a Japanese signal. That's really what it boils down to. And some of these other loci were ones that were found for heart disease, but now we're suggesting that maybe they might be more specific for MI. So that was kind of like a whirlwind tour of how we're approaching this sort of like, at least trying to make a contribution to the genetic architecture of cardiovascular disease without really competing with the big groups, right? Trying to sort of like find our little niche. And I told you that the paper that we published last year, basically the author group doubled in size from what we first when we first drafted the paper. But in the end, and this is what I wanted to tell the students, that this optimism is like, well, yes, you get batted down once or twice, but in the end, I think we got a much better paper out of it, quite frankly. So sometimes, getting rejected doesn't have to be a bad thing. You get to work with a bunch of other people, and you end up with a better product. But I specifically wanted to sort of highlight some very good collaborators that we've done more with. People like, in my group at USC, especially Yana and Yi. Yi was a former postdoc who did a bulk of the work on the MI analyses. James is a new student who's kind of carrying it on. Zhang is a former student who's now in a PhD program. All the consortia without whom, we couldn't do this, providing their data either publicly or through collaborations, things like that. My longtime collaborators and former mentors at UCLA, at the Cleveland Clinic, and then U of A and University of Virginia. So thank you very much. And I'm happy to take as many questions as you like.
Wow, thank you. So questions for Hooman?
Can I ask a question?
Sure.
Thank you for interesting talk. I'm wondering the SLC44A2 is also expressed in the those coronary artery tissues.
Yeah.
I'm just wondering if the 44A3 is actually localized in mitochondria, or maybe the cell membrane to traffic the actually choline to the inside of the cells, and then
Yeah.
A2 might be just trafficking to the mitochondria, or both of them are localized in the mitochondria and compensating each other. I'm just curious, what do you think?
Yeah. So that's a really, really good question. So unfortunately, I'll tell you, 44A2 is expressed in the artery wall as well. So that's the answer. I mean, if you do the same sort of single cell RNA-seq sort of analysis using that website PlaqueView, you'll see 44A2 expressed in the artery wall. So it's possible that it's also contributing, right? So that's the answer to the first question. The answer to the second question is there is no good antibody, like I said, even for in situ, to determine whether it's localizing to mitochondria. So what we were thinking of doing is just basically making like a GFP tagged SLC, and then looking to see if it goes to the mitochondria or where it is in the cell. While we're at it, we might as well do the same thing with 44A2 and show that they're both in the mitochondria, right? Especially in smooth muscle cells, artery wall cells. Yeah.
Thank you.
Mm-hmm.
Hi, Hooman. I do have a couple of questions. Great talk. It was like I learned a lot actually. So I have two questions. One is a very naive question about stratifying MI patient, right? So these are people that do not maybe have CAD, but MI could be because of heart, like, conditional, some sort of condition in the heart muscle. It can be, for many reasons. So I'm wondering if you have a way of stratifying that, and if stratifying different type of MI increases your signal for your locus that is maybe specific for one type of MI.
Yeah. So most MIs are because of atherosclerotic plaque rupture. There are a small number of MIs that are like from coronary spasm or things like that, but those are sort of not obviously as prevalent as the atherosclerotic, just a typical cholesterol buildup, et cetera. Unfortunately, whether for at least access to the data that we have, which is the UK Biobank, there isn't really a way of determining, but we did exclude anybody who had an MI because if they had like sort of coronary spasm or something like that, right? You can have basically no plaque, but your veins or your arteries sort of like spasm, and that causes an MI as well. Basically, MI means like lack of blood flow.
And this is the reason why I'm curious if there is any correlation of your locus with something specific, like, some sort of cause.
Yeah, so we don't have, maybe the only thing that we might be able to do is use the angiography cohort that we have access to with the primary data to kind of try to figure out what kind of MI there is or there isn't, but that will dramatically reduce the sample size to only like a few thousand compared to, but it's a very good question to determine what kind of MI does this predispose to.
Yeah, and then I have another question, a kind of independent question. So at the beginning, you showed us that this gene overlaps with a link, right, with a non-coding RNA.
Yeah.
And if I remember correctly, your SNP is within the non-coding RNA.
Right.
Right. So have you tried to measure the expression of that?
Yeah, no, we haven't. And it's actually, it's called a link, but it turns out now the annotation is it's an antisense to SLC44A3.
Okay, but so, it is an antisense non-coding RNA?
Correct.
Yes, right?
Yeah.
So you should have the transcription of that, like you should have the transcript. And then, maybe, I'm thinking if that has also a sort of like function, because it's an antisense, so it's possible that is highly expressed and you get down regulation of your gene.
That's right.
But it's possible also that your link is regulating other genes.
Yeah.
So maybe it's not necessarily the effect of the SLC, but also the link that is associated to it.
Right. So I will tell you that we haven't specifically quantitated ourselves, but I will tell you that in StarNet, that cohort we used to do some of the functional data, it is measured, that antisense is quantitated, but we don't have an EPTL for it with our SNP. So it kind of suggests that maybe it's not working through the antisense, but I think we need to do a better job of really figuring that out.
Very interesting.
You and many other people, every time we show this that regional plot, immediately say, "What's with the non-coding RNA?"
Yeah, I mean, I'm obsessed with that. So
Yeah.
because of the 9p21 and and Ariel, but no, I think it's a beautiful story, and I think that it's very, you used a lot of different cohort and a lot of different databases, which is like something that I haven't approached yet, but I think it's super powerful.
It's because we're a small operation, we end up being good at doing sort of like this bioinformatics. I don't have.
But it's great because I think that this is how you use this type of big data set. Otherwise, there's they sit there and they don't do that much.
I'll be very honest with you. You know what has been really useful? It's Google search. Like when I wanted to figure out if SLC was 44A3 was expressed in platelets, I'm like, where am I going to get that data? I just basically just said RNA-seq platelets, and that paper came up out of nowhere. And then you went into the supplemental table, and sure enough, they had all the transcripts out of their RNA-seq data. I was like, well, how hard could it be to do a Control F and find SLC 44A3 and A2 in their list of things? So it wasn't like something sophisticated. It was just basically Google searching. So
I think that this is very useful for the student, that sometimes they think that, how am I going to find this? The Google search.
I still to this day to my students are like, oh yeah, I looked, and I'm like, I couldn't find it. I'm like, did you try a Google search? Just Google search. You never know what you're going to find. I mean, it's so easy. So yeah.
But thank you. Beautiful talk. Thank you.
Oh, thank you very much. Thank you. What about the students? I know that they had a lot of questions. I don't know if I answered all of them.
I think maybe this is the time to allow the students to take over.
Okay.
Right,
basically, it's just the students and the few of us left. So go ahead, students.
Fire away. Is anybody there?
Oh yeah, they're there.
I was wondering, so your data, it seemed like you talked some at the beginning of your slides. I know you talked some about looking for risk factors and some about like parsing out the pathophysiology. What do you say your findings? Would you use them more for screening patients for risk or preventive, or more for treatment, or just whatever the scientist picks them up decides to do?
Yeah, that's a good question. So can we stratify risk based on like, let's say this SNP for MI? I don't think so, because if you look at the odds ratio, right, the effect on how much it increases risk of MI, it's very small. And obviously it took like 600,000 people to find that effect, right? So I'm not sure if it's going to be really useful clinically to like sort of stratify people into high and low risk. For drug development, it's very premature, I would say, although that would be like the great thing, because really we don't even know what it does. I think we'd have to sort of like characterize the gene a little bit more, and then you'd like to have, presumably, if everything works out, you'd like to have like an SLC44A3 inhibitor that maybe only works in the vascular wall, right? So you give it to people with heart disease and you say, okay, this should reduce your risk of MI, presumably because of its effects on mitochondria. That's a pie in the sky kind of thing too. But the last thing you brought up, I think, is maybe it will give us some idea into the pathophysiology of MI. And we can sort of like explore that a little bit more, whether it's through smooth muscle cells or another cell type, et cetera. And I think with the mouse, especially if we can show the mouse has reduced thrombosis, then it would be a good model, to sort of pursue from a functional perspective. So I think of the choices you gave me, Grace, I would go with door number 3 as the one that's most useful.
Thank you.
Mhm.
So Hooman, isn't it critical for you to figure out what cell type is the one that's mediating
Yeah.
this
Yeah.
thing? And aren't you going to knock it out in some of the candidate cell types? Are you going to knock it out in smooth muscle and macrophages and endothelial
Yeah.
cells, et cetera?
Okay, so Alex, so there's a backstory to that too. When we got the mouse, right, we got the mouse that was the global knockout, and we also got the mouse that was floxed, precisely with the idea of, okay, if we have the floxed mouse, now we can start making cell type specific. So the mice that they sent us from Germany were 6 months old. And these mice, both lines, like both the global knockout and the floxed line, sat in the vivarium for 6 months, and we tried everything in our power to get them to breed. Right. And nothing was happening. And I'm like, I came to the point where I'm like, you know what? We're just not going to get any pups. So then, right, like literally a week before, I said, okay, let's take the remaining male mice and just basically cryo freeze the sperm, and we'll come back to it later. We got 2 pups from the global knockout. The floxed line died off. So we would have to get the mice all over again. I told you about the import fees and all that kind of stuff. And I'm not sure we want to do that. But we have another way of doing it is there are endothelial cell lines, right? And there are smooth muscle cell lines. And of course, there's like, THP1 cells or whatever, so that we can overexpress SLC in vitro, doing it that way, right? We won't be able to recapitulate the sort of the MI phenotype, but we can at least look to see if mitochondrial cell or DNA content or function is altered in a cell specific way that way. That's the idea. All of this is in the grant that we proposed, so.
Yeah, I want to talk to you more about that some other time.
Yep.
I noticed in the review that one of the mechanisms that you talked about that could be an important influence in like MI and CAD was like the microbiome. And I was wondering if your lab had any, or like anybody you know had any intention of doing like a wide scale microbiome analysis.
Yeah. Andrew, we're not doing microbiome directly ourselves, but there are a lot of people that are interested in microbiome and cardiovascular disease. Certainly microbiome is involved in a lot of things, right? I mean, probably the best one is like, intestinal diseases like IBD or, whatever. But with respect to cardiometabolic, I would say like, Stan Hazen at the Cleveland Clinic, who we've worked with a lot, is probably one of the leaders in this field. And he's identified a lot of metabolites that come from the microbiome and influence disease risk. But we ourselves really haven't done any microbiome research ourselves. We're more interested in like maybe finding the genetic factors that are associated with microbial products, like these metabolites and things like that. And I'm not sure if it was your question or somebody else's question, but kind of related to that is Stan has showed that you can target the microbiome pharmaceutically and sort of decrease atherosclerosis in mice. So as an extension of what you're asking is, well, if the microbiome is involved in heart disease, can you target the microbiome as like a way of like, therapeutically? And the answer is possibly, yeah. But I don't know if I answered your question completely, but yeah, that's.
I think that the most interesting part of your answer was the idea that a certain microbiome can have a different influence based on the genetics of the organism that it's inhabiting.
Yeah.
In terms of like influencing CAD. So
yeah,
thank you.
Sure. Does anybody work on cardiovascular disease?
I do.
Elsa.
I'm Valentina's grad student.
Okay. All right.
Are there any more questions?
Yeah, let's ask questions.
I have a question. So last week we had a presenter talk about like genetic interactions and stuff and like imputing data to kind of raise statistical power to detect this kind of stuff. Like, has anyone in your lab like considered like doing like taking that kind of like a network approach to seeing if like, because you talked about like the like missing heritability, and like
Yeah.
last week something that we talked about briefly was like, you know, genetic interactions could be contributing to that missing heritability. And I was
Yeah.
wondering if, like, you know, with like the sample sizes you have where like the cohorts were like so large, like has anyone considered like looking at those kinds of things?
Yeah, right. I'm sure people have thought about it. The problem is this, you know, like we ourselves haven't really done a whole lot of gene-gene interactions. We have, because we have access to UK Biobank data, we've actually started doing gene environment interactions, meaning like, you know, can we do a gene air pollution interaction? Can we find genes that interact with air pollution to affect disease risk? So that's not really the network kind of analysis that you're asking. You're looking for like a network of interactions between genes and things like that.
Yeah.
And no, we haven't done that ourselves. I think that's probably a little bit more computationally challenging for what we do. The other problem with it is that even with a sample size of 500,000 people in the UK Biobank, my guess is that you will be underpowered. And without getting access to the primary level data from the other cohorts, which we don't, like you can't apply and get data to Biobank Japan. UK Biobank is very unique that way. So I think you're going to need larger sample sizes to do that. And I'm not sure with even 500,000 people, it'll be good enough. So yeah.
Cool, thanks.
Mm-hmm. Who has a question?
Hi, so as a follow-up to that, I have a broader question about like the perspective of these kind of data, these biobanks going forward in the future. Because like we know that not only do you mention that there's not enough power even with that amount of people, but also that like the diversity of these populations are also not that broad, because like, for example, in the biobank Japan, we got that SNP that was only for the Japanese population. So I'm just wondering, like, broadly speaking, for human genomics in the future, are there like more of these cohorts, more of these biobanks coming out soon, and will they be like more sufficient for these genome-driven research?
Yeah, I think if I understood your question is like, you're wondering, can we sort of apply this to other ancestries, right? Or like other groups? Is it gonna be improve, or can we improve our understanding by expanding to other ancestries? Is that what you're asking, Wei?
Yeah, partly that from a theoretical level, but also like I want to know about what is the current status for these kind of database as they are being developed in like all the populations in other countries as well.
Yeah. Okay. So the biobanks that I'm aware of that are large, obviously, is the UK Biobank. There's Biobank Japan. There's a big one in China. There's also a one in Finland, which really is sort of like another. Well, it's European ancestry, let's put it that way. But in the US, there's what's called the Million Veterans Program, or MVP. And it's basically taking advantage of all the sort of war, or people who are in the Veterans Administration system. It's basically a cohort of patients, right? And because of that, you do have a substantial number of African ancestry and Latinos and things like that. So that biobank could be useful for genetic studies. And in fact, I mentioned to you that, like, you know, as of now, there's like 200 loci for heart disease, but I do know that a couple papers are coming out where they've used the Million Veterans Program and they've found like another 100 or so genes. So now like the number of genes for heart disease is going to go to like 300 or so because of those other cohorts. I'm sure that there's other ones like in Africa itself for people of African ancestry. There's other ones in South Asia, et cetera, but not quite the size that, like, let's say UK Biobank is. Some of them will be publicly available, some of them won't. So it kind of remains to be determined how much we can get access to those.
Thank you. That's a very interesting prospect for us in the near future, at least.
Yeah. Yeah. A Google search might help. Yeah. Liz, you had a question.
Yes, I do not remember if it was the paper itself or the review, but they say, so they were talking about so they made an observation that some of the mechanisms that establish, and I'm gonna butcher this word, atherosclerosis, or that its progression may be different from those of the predisposed plaque and the thrombus formation. Have you looked at those, like, have you been able to identify which of those mechanisms has like the stronger correlation, and are you interested in like maybe like mimicking those mechanisms in like maybe mice to see
Yeah.
how it works?
Yeah. So, you know, I mean, people think let's say that you have high cholesterol, right? From whatever, middle age, young age, whatever. And the high cholesterol basically gets into the artery wall and the plaque kind of grows. And because the plaque grows so big, then eventually it ruptures and you have a heart attack, right? That's the standard, like, sort of understanding. And yes, that's true. You know, as the plaque gets bigger, it's just more prone to rupture. But there are some people who live for 80, 90 years with heart disease, and yet their plaque never ruptures. Why is that? Right? Why is that? Is it because their plaques are sort of like harder and they don't rupture? And if that's the case, is it because of their genetics? And so that was the whole purpose of why we did this study, right? It was like to find somebody might have a smaller plaque, but then carry like a bad gene like SLC that might actually make them have a plaque rupture, right? Even though their plaque is smaller compared to somebody who's got like a really big plaque, you know? That's kind of like the idea. And I think what I was showing you at the end was to say, can we do a broader search and find more of these genes? And I think the answer to that is going to be yes. I don't think we're going to find 300 genes that are going to be specific for MI. It's going to be a small subset, but I do believe that there are some genetic risk factors out there that could be clinically more important, because those are the people, you don't want people to have a heart attack. I mean, if they have heart disease, you can live with that, but you just don't want them to have a heart attack. So, yeah. I don't know if I answered your question, but I tried.
Answer my second question, which was going to be like, do you think that genetics has a big effect when it comes to plaques and heart attack? And I'm asking because, so my dad, right, he had a heart attack, multiples of them, right? But he's just a strong man. So thank goodness nothing ever happened. And the last time they went in and they checked and he had plaques, right? Mm-hmm. But the doctors were like, your dad has had this since he was like young. Like he has this since he was super young, but it never affected him until now that he's a little bit older.
Yeah.
Like, do you think genetics has also like a big role when it comes to how much it affects someone?
Well, you know, you can ask in your family is like, did your dad's dad or your dad's mom have heart attacks? I mean, that's like one of the first things. You know, my dad had a couple heart attacks too. And then when I look at my family history, I'm like, oh yeah, okay, you know, his dad had a heart attack too, or like, you know, his brothers had a heart attack, you know, et cetera. So clearly genetics has an important component, right? In some people, it's more important than others. In some people, they might actually not be genetically predisposed, but they're a heavy smoker and they eat really bad. That can also cause a heart attack, right? So yeah, I mean, clearly genetics has a strong component to both heart disease and MI, right? So yeah.
Okay, thank you so much.
Mm-hmm.
I wanted to ask if like you know anything about like sudden lifestyle changes causing the breakage of the plaque. Like, for example, I knew somebody that was they were a smoker, they would smoke cigarettes, and they got pregnant, and the doctor told them, actually do not quit smoking right now so suddenly, because it's the stress of quitting smoking that can cause a miscarriage, as opposed to like the danger of continuing to smoke cigarettes while you're pregnant. And so like that's why I'm wondering like if like a sudden lifestyle change could cause the breakage of that plaque that somebody might have had their whole life.
You know, I think generally speaking, it would be probably stress, I think, is one that might actually cause, you know, like if you want to call like acute stress a lifestyle change, that can certainly be associated with a heart attack. But another one is if you look in regions of the world, like China, for example, or other areas where like air pollution levels spike for whatever reason, right? That's associated with increased risk of heart attacks and stroke. So I know it's not a lifestyle change, but it's an environmental sort of exposure that in an acute setting can also cause a plaque rupture for whatever reason, whether like having like, you know, really high air pollution increases the inflammation in the blood and then that causes the plaque to rupture, or stress kind of increasing stress hormones and that causes the plaque to rupture. I think those are maybe a couple examples that come off the top of my head. But otherwise, like, oh, maybe like if you acutely start exercising, like if you start running, like, without having trained or anything like that, that can certainly cause a problem, right? So yeah, there's some examples of lifestyle changes, but I don't know of a lifestyle change where getting rid of something bad actually causes something bad. So, no.
Hi, Hooman. Thank you for a great talk. I had a question. It's kind of more a broader question, but I was like wondering, it kind of ties in with Hui's question as well. I was wondering when you use big like biobanks and data sets like this, I'm not sure if this applies to CAD and MI, but I know that different populations can manifest symptoms differently.
Mhm.
So, how can you take that into consideration, considering there might be a different mechanism at play in these different populations without also limiting your data set too much where you're
Yeah.
missing things that are common across all of them.
Yeah. Again, if you wanted to sort of harmonize your outcomes is what you're kind of getting at, I think you would need to get your hands on the actual raw data itself and say, okay, this is kind of what Valentino is asking, like not all MI is the same. Can you stratify, can you sort of stratify people into this group or that group? It's hard because I don't know of anybody that has access to the raw data across all these cohorts, right? So the Biobank Japan people probably obviously have access to their own data. They might be able to get access to the UK Biobank, but not the other cohorts, right? And for us, we got access to UK Biobank, but we don't have access to the Biobank Japan. So we are going to be limited. And extrapolating our results to these other cohorts will sort of only capture the genetic signals that are comparable, but we could be missing a lot of things because, MI is not the same in Japan versus, the UK or whatever, right? So yeah, I mean, there's certain the granularity of these kinds of analyses can't be done as like you suggest in all cases. So you're just limited to what you got. But you know what? Given that we didn't really spend a whole lot of money to get all that data, we'll take it with its caveats. Yeah.
Sure, that makes sense. Thank you.
A conversation that we had that was really interesting when we did our first class about this information about your papers and stuff was as to whether a strong degree of plaque breakage might actually be like an evolutionary benefit in terms of the plaque breaking off before it becomes very large. And so what do you think about that?
Maybe. I don't think so. I think every time, basically a plaque rupture is essentially like a cut, right? It's a cut. So what happens when you cut your skin? The body immediately tries to cut off the blood flow, right? And that's why you get a clot and you stop the bleeding. So a plaque rupture is essentially like a cut, but it's on the inside of your body, right? And that's why the clot occurs, because your body's like, oh my God, I'm bleeding internally. I got to shut it. I got to close it off. And so when you do that, that's where the thrombus comes from. So if you have a small plaque and it breaks off, does that mean that it gets rid of the whole plaque that was there? No, it basically means you scar over what was there, right? So I don't think that, evolutionarily, it would be advantageous to start rupturing plaques early on to get rid of them.
Okay, thank you.
I had a question about this, which you were mentioning with how SLC 44A3 doesn't seem to show association with other CAD risk factors or with a whole bunch of these other things that you might expect it to be seeing, having some interaction with, which seemed just so interesting to me when I was reading this paper. And I apologize if you already answered my question. I did have some internet connections partway through the meeting, but I was wondering, where are you thinking of going from here? Is this something that you think just doesn't really interact with other factors for coronary artery disease? Are there other avenues of research that you think could reveal interactions with other factors?
Yeah. So Elsa, the big question is, okay, if you believe, first of all, that SLC44A3 is the gene that's predisposing to plaque rupture, the big question is, what's the mechanism? What's the mechanism? So like you said, we don't think it has anything to do with blood pressure, with lipids, with diabetes, with obesity, none of the traditional things at all, right? Not even metabolites or anything like that. So what I think it's doing is it's actually affecting vessel wall function. And we don't know which cell type it's affecting vessel wall function, but at least if we had to prioritize, we think it might be smooth muscle cells. So for whatever reason, genetically, altered SLC function changes smooth muscle cell function, possibly through mitochondria, and that somehow destabilizes a plaque that has grown for other reasons, whether it's cholesterol or something like that. So once you have plaque for whatever other reasons, then if you carry this SLC variant, we think it might actually alter your smooth muscle cells such that it predisposes your plaque to rupturing. Now,
and you said that, sorry,
go
finish.
ahead. No, go ahead.
I'm just really, really interested by it because you said that there's no interaction with 9p21, right? Is I got that correct? Or there's no evidence for it?
No, I mean, like, I think what I wrote about 9p21 was like chromosome 9p21 is both associated with the presence of plaque, meaning heart disease itself, but then you also look and it's also associated with MI too. But the reason it's associated with MI is because it's associated with CAD, right? It's kind of like contributing to the development of the plaque, and then it's also contributing to plaque rupture. That's an example of a genetic factor that's associated with both, right? But SLC is one that seems to be specific for MI.
Just MI, okay.
Just MI, yeah, just MI. And that's where I think is, at least for us, it's kind of a very interesting concept that maybe, we can, like I said, as I've mentioned before, you can live with having a plaque for a long time as long as it doesn't rupture. So if we understand the mechanisms that promote rupture, then we intervene there, as opposed to just preventing heart disease from the first place, which I don't think we're going to be able to do completely.
Yeah. And so you're thinking it might be changing something in the structure of the vessel. Okay.
Yeah.
That's really, really interesting because we're looking at adhesion and contraction functions in SMCs in vessel walls in connection with 9p21.
Right. So I mean, then a lot more about this than I do. Is like, how could how could SLC participate in smooth muscle cell function such that it only predisposes to MI? What is it doing? If it is even smooth muscle cells, I can't even tell you for sure if it is or not.
Of course. Thank you.
I might have information from the beginning because I come a little bit late, but I was wondering, by the mechanism of heart attack and CAD, is it possible to have heart attack without having CAD? This question is, it's related to a second question, like what would be the challenges to define CAD specific loci as compared to, define MI specific loci? In your experiment, it's kind of not that hard to control for CAD plus with MI plus or minus, but it seems hard to do the other way, to control for MI plus and do CAD plus and minus.
Yeah, I will tell you, this is kind of what Valentina brought up. Most MIs, most heart attacks occur on the background of some sort of heart disease, right? There are rare cases of actually having, like I said, coronary artery spasm, where it just sort of like constricts and it cuts off blood flow, but it's not because a plaque ruptured and then you got a clot that closes off the artery, right? And those mechanisms are, I think, different than what we're talking about, right? Those could be sort of like vasoconstrictive mechanisms and things like that, like, like basically a muscle spasm, so to speak. Maybe actually smooth muscle cells, because that's what causes the spasm. So, but there are so few of those, right? And if those happen, they probably occur in younger people. Like if you heard of like a 30-year-old having a heart attack, unless that person had a really, really high cholesterol levels, I would have a hard time imagining that that's because of atherosclerosis. That's probably because of some sort of spasm or something like that. Unfortunately, it's hard to find those unless it's written in the chart somewhere saying this person had a coronary spasm versus, versus atherosclerotic MI. So I'm not sure if we're ever going to be able to address that question suitably. At least not us, because we're not, I mean, I'm not a cardiologist, so I can't even get those patients if I wanted to, right?
Yeah.
So, yeah.
Sure, thanks.
Mhm.
I have a question. So it sounded like mice generally don't have plaques unless you do that tying off technique that your collaborator does. Do you worry about testing this gene's effects in mice on a situation where maybe the plaques are generated in a totally different, via different mechanisms? That's the first question. And then the second question is, are there alternative animal models which do form plaques and do have heart attacks in which you could maybe artificially increase or decrease SLC 44A3 levels in the relevant cell types once you know what the relevant cell types are?
Yeah, so Jerry, answer to your first question is you can make mice very hypercholesterol. You get cholesterol levels like above 1,000. There's like ApoE knockout mice, LDL receptor knockout mice. You can get like cholesterol really high, and they will get plaques developed. Like you get extensive atherosclerosis. The problem with mice is that those plaques never rupture, right? So even though they have like, atherosclerosis up and down their like sort of aortic branches and tree, they won't rupture and cause a heart attack. Like a mouse will never keel over from a heart attack, even if it's got atherosclerosis everywhere. And so you do have to use these surgical sort of constriction models to kind of mimic the ischemia that human arteries develop. And then that does, in certain cases, like I showed you, cause a plaque to rupture. But even so, even if you do that surgical model in mice that have extensive atherosclerosis and have really high cholesterol, it's maybe at best 50% of the mice that have undergone the surgery will actually have evidence of rupture. The other 50%, it won't do it. You know what I mean? So it's not a perfect system by any means. So are there other animal models? Maybe you could use pigs. People do use pigs for certain coronary, and atherosclerotic things like that. But to go and do a plaque rupture model in pigs, we're certainly not equipped to do that ourselves. And I think we would probably take our chances with this surgical model in mice, just because you can at least do like 20 mice in each group. I don't know how many pigs you would need in each group to do that.
Yeah.
But there is a lot of controversy in the field about, some people just like say, oh, that model for surgical model for plaque rupture is doo-doo. And somebody else says, no, you got to use this other model. And somebody says, no, you got to do this. So I will tell you this, if somebody comes up with a good mouse model for MI, they'll make $1 billion, because every drug company is going to use it to show that their drug reduces MI. Right?
Do other animals have MI? I mean, do pigs have MI? Do they get MI?
That's a good question. Yes, I think pigs do get MI.
Okay.
Larger animals get MI because in mice, the coronary arteries are kind of small, obviously, right? I don't even know if a rat can get an MI. So it's mostly maybe a large mammalian disease or larger mammalian disease. Yeah.
Can I ask you a general question?
Of course.
I really liked that you share the development of your paper.
Yeah.
Actually, I enjoyed at the same time, I kind of share the pain, the same pain as you had.
I feel your pain. We feel your pain.
Kind of ouch. But I really liked how you developed the project in that way. And I think I need your trait to get optimistic. But I think because of this, I think you had a kind of interesting niche that can be applied to any disease, that you can actually identify the genetic factors now to look at the development of the disease in the different points. So that finding itself kind of gives the mechanism of how the disease is developed. So do you have any idea that other disease that you can think of to apply that your methods to, to understand some of the development of some particular disease?
I suppose any disease where you can stratify subtypes based on pathology or something, like not everything is, and you could do this analysis for. So for example, like this is not my field, but like ulcerative colitis, right? There's Crohn's disease, there's, IBD, there's different, one is a little bit like upper in the GI, one is lower. So what if you kind of like, stratify that way, right? I could kind of think maybe, and I'm sure this has been done, where there's different genetic factors for one type of intestinal immune disease versus another.
Maybe. Yeah, maybe you can actually look at the genes that activate the immune system, and maybe that genes that actually the causing the damage. That makes sense.
And then another way you can possibly is, for example, like I'm just again thinking off the top of my head, like diabetes, we call it diabetes, which is dysregulated glucose. But that can come about either because your beta cells in the pancreas give out. This is obviously something Alan knows very well. Or it could be because you're insulin resistant, right? And so if you can take diabetics and stratify them and say, oh, these are the beta cell failure diabetics, these are just the insulin resistant diabetics, and then do genetic analysis when you've sort of stratified patients, do you find different genes, right? Yeah. So that's sort of another thing. You could probably apply it to other things. Cancer, it's actually probably a very good one, because cancer, you can profile tumors and stratify into different groups.
I think.
Right?
Yeah.
So.
Yeah, it was a very good example in that sense too. Thank you.
And this thing about like, the journey to getting the paper published. I teach in a class where we teach our first year graduate students, our PhD students, like, how do you actually get a paper published? And the reason why I decided to put it for this talk was because the students were on, and I do this for the other class too, to just say like, it's not an easy path. You have to sort of overcome, et cetera. And I just don't give up. I just don't like to have somebody tell me no. I think that's really what it is. It's not like pathological optimism. It's maybe pathological refusing to take no for an answer.
That's actually really great.
I mean, if it's going to be no, I'll say no, not you. Or that's at least that's what I keep telling NIH. If it's going to be a no, then I'll say no, not NIH.
Very great.
What is it, like 5:30 you guys' time?
Yeah, 5:25. Anybody else? Why do we?
Okay.
All right. This was so much fun. I really wish I was there in person. Maybe next time.
Next time. Yeah, I really enjoyed. Thank you so much.
That was a great talk.
Thank you for a great talk.
Yeah.
Glad you guys liked it.
Really good.
So let's keep in touch.
Yep.
Okay.
All right.
Okay.
Take care now.
Thank you so
Bye,
much.
everyone.
Thank you.
Bye-bye.
I'll let you know if flies get heart attacks.
Okay. All right, Drosophila. Great model. Large sample size.
It has a heart at least.
Yeah. Take care. Bye-bye.
Bye.
Bye.
Thank you.
You're welcome.